Good morning friends, welcome back to another very short video tutorials on biology the brain buffer. Today we shall be talking about one of the most important part in the plant physiology, photosynthesis, C2, C3, C4 cycle along with the crassulacean acid metabolism. So in the plant kingdom there is a huge diversity as you have different domain and geographical variation like tropical area to the savanna. So depending on those we have different types of plants. So I shall be sharing my PPT with you. So stay tuned and join. Okay, so let's begin the discussion. So basically, as we know, there are three different types of plants, C3, CAM and C4. So basically, after the capturing of the gas CO2 from the atmosphere, if the first stable product contains 3 carbon, then we call them as a C3 plant. If the first stable product is containing 4 carbon, we call them C4 plant. And if it is the first stable compound is with 2 carbon during photorespiration, we call them C2 plants. And CAM is the one of the important and unique feature of acid metabolism in crassulacean group of plant. So crassulacean acid metabolism is a very specific method of photosynthesis in succulent plant. So let's begin with it. So basically, if you take the cross section of a leaf, if you take a leaf and take a section of a leaf, you can see oxygen can be captured, sorry, CO2 can be captured and entered through the stomata. So basically I am drawing here the palisade parenchymal cell, and you have a spongy parenchymal cell in the lower part of the leaf, and you have a stomatal pore through which the CO2 will move inside. Now in the mesophyll cell, mesophyll, it's a parenchyma living cell with chloroplast where the Kelvin cycle will be continuing. So with the entry of the CO2, the final product is glucose. Remember, the C3, C4 and CAM all destined to create glucose. Later it will be formed sucrose and starch as a storage. But as you study the C4 plant, here you can see there are two different types of cell, mesophyll and bundle of sheath. So mesophyll is common for C3 and C4, but this bundle of sheath is extra. Now, if you study the anatomy, later we will be talking about it. Simply you have a mesophyll cell and it is surrounded by a bundle of sheath that is very thick. And this particular type of anatomy is called as a Cran's anatomy in C4 plant. So due to the presence of Cran's anatomy, you can see Kelvin cycle take part in the occurring in the bundle of sheath. Then finally glucose is formed. And in CAM plant, what happens? At the night time, it is captured in a form of an acid. So it could be oxaloacetic acid. Later we'll be talking about in details after some time. And same way, the daytime mesophyll Kelvin cycle continues and glucose is formed. Melvin Calvin got Nobel Prize for the discovery of this beautiful cycle. Now the question is that why? Why these three types of adaptations are present in plants? Basically, like 85% plants are C3 plant. So in normal condition, all conduct this C3 cycle where the first stable product is PGA, phosphoglyceric or phosphoglyceric acid. But there are two extreme conditions. One is like high temperature. Like some plant have to sustain or survive in a high temperature region. For that purpose, the specific adaptation is C4 or C4 plant take those kind of adaptation. But in case of a CAN plant, if the place is very dry, so they will try to conserve water. So what I am saying, there are two purpose. One, if you want to conserve water, then you should take the C4 path, CAM pathway, CAM pathway or crassulacean acid metabolism pathway. And if you want to survive in high temperature, then you have to go for a C4 cycle. So C4 plants are basically designed to tolerate high temperature, and CAM plants are basically designed to reserve or conserve water so that they won't lose water in a dry place. Now, if you compare in little bit statistically, 85% are CAM plants. Now, if you take it like a 2 to 3%, I think you can see here. Okay. So now 8% of the entire distribution in the plant kingdom, if you study, let me check whether recording is, yes, it's going on. So 8% of the entire plant kingdom, that is the CAM. So statistically 85%, the most is a C3, then some are CAM, and some cases you can see the C3. Okay, it can change little bit in the different places, and type of photosynthesis in different this condition. You have a C3 photosynthesis where the first stable product is PGA, phosphoglyceric acid. Here first stable product is oxaloacetic acid. In CAM, what they does, they simply capture the CO2 at the night in the form of oxaloacetic acid. So some part of the CAM is like a C4, and later they can convert it to the malic acid. So I will be sharing a better picture with you. So now if you study the leaf anatomy, you have this kind of spongy parenchyma here below, like a sponge like structure and like sponge in fiber, and you have a palisade parenchyma. So this is how the things actually look, but it doesn't have any cranch anatomy. But if you study the C4 plant, they have a special arrangement of bundle of sheath, that is in a form of cranch anatomy. So you can study here as a cranch anatomy is present, and thick and fleshy leaves. As you know, the succulent leaf, mesophyll cells having a large water-filled vacuole. This is the property of a CAM plants. The stomata open in the daytime and close at night. Very normal condition. Stomata open at the daytime and close at the night time. So this is the similarity between C3 and C. An inverted stomatal cycle, like it's open at the night time and close at the daytime. And you can see the temperate, most common, and tropical and semi-tropical region, the high intensity, high temperature, high light intensity. Favorably C3, C4 plant grows the savanna in that place, and in the desert or those very conditions, the CAM plants will survive. Now, if you study the anatomy of the leaf, you can see here that a bundle of sheath, veins and stomata, the mesophyll cells are over here. You see the palisade parenchyma, and you have underneath the spongy parenchyma. These are the parenchyma cells present here. But here in the C4 plant, you have a circular presentation. At the center, you have a bundle of sheaths and vein, and the photosynthetic parts are over here. Or you can say you have a xylem and phloem tract, then bundle of sheath cells are present over here, and that one is a very thick. Then you have a other photosynthetic part. So if you take a close overview of it, what is the similarity between all these three? The most common part is the presence of enzyme, that is rubisco. Rubisco is present in all these three cases, C3, C4 and CAM. But you know, rubisco is a ribulose bisphosphate carboxylase oxygenase. So carboxylase function is present here, carboxylase. At the same time, it can perform as an oxygenase that can bind oxygen in a certain condition, like the high temperature, high light intensity and low CO2 concentration. In that case, the C3 plant will be converted to C2 plant. That is the problem of having rubisco. Now, if you take it like C3 and CAM, what is the similarity? You can see Calvin cycle occur in mesophyll cell. So Calvin cycle occurring in the mesophyll cell for both C3 and CAM. It's normal for it. But remember, Hatch and Slack pathway is totally absent in C3. So Hatch and Slack pathway or C4 pathway is common. You can see this part is common for CAM and C4. This is the beautiful representation. So Hatch and Slack pathway is present in CAM and as well as C4. So this is the most important part. Now tell me what is not present in CAM and common between C3 and C4. You can see production of glucose. See, CAM also try to produce the glucose, and it is mainly in the crassulacean plant. And stomata are open during the daytime. This is the most important part. So the stomatal opening, the stomata related issues are there in the CAM plant. So similarity between C3 and C4 will be connected with the stomata opening timing, that are same. Photorespiration seldom used, because in C4 plant, you know, this photorespiration hardly take part because the most abundant and dominant enzyme is a PEP carboxylase. So that is working to fix the CO2. So as a substrate level in C4 plant, if I write here, as a substrate, there is a phosphoenol pyruvate molecule, and it can capture the CO2 gas in the presence of PEP carboxylase enzyme. Then you have a C4 compound, that is oxaloacetic acid. So they don't have a fear of photorespiration, as Rubisco is not that much working at that moment. There are some more differences. If you are interested, you can note it down. Like occurs in all plants, occur in 3% of the vascular plants, and some other examples. Now the initial CO2 acceptor, this is the very important. For C3 plant, the acceptor is a RUBP. Now after the acceptance of CO2, they will convert it into a PGA, that is phosphoglyceric or phosphoglyceric acid, depending on the group. If it is C++, then you can call it as a phosphoglycerate. Now here the first stable product is oxaloacetic acid in case of a C4, because it contains also 4 carbon. You have also studied this one in the Krebs cycle, and the first stable product in CAM is a 3-PGA as C3 plant, and at the daytime, and oxaloacetic acid at the night time. But remember, in C2 cases, when photorespiration is going on, then phosphoglycolate that contain 2 carbon. So phosphoglycolate along with PGA, it is formed and it contains 2 carbons. Next, this is the molecular electron dot structure of the Rubisco enzyme. So it is the most abundant enzyme and protein also in the plant kingdom and the world also. In the world, it is the most abundant protein enzyme. Now let's see the dimorphic chloroplast arrangement in the C4 plant. You know, there are mesophyll cell and bundle of sheaths. So bundle of sheath is here. You have xylem and phloem, so vascular tissue, then bundle of sheath, then that is surrounded by mesophyll cells. So you have 2 different form of chloroplast in mesophyll and the bundle of sheath. That is the condition specifically found in C4 plant called dimorphic chloroplast, like corn and ziames. Now you have the beautiful picture. So as CO2 is get captured, it will run Calvin cycle in rice, C3, all C3 plant. You take mango and other all important plants. So they will finally produce the glucose, and that will be converted to sucrose, then starch. Sucrose is reducing or non-reducing sugar. Sucrose is, anybody is going to tell me? Glucose is reducing. So now you don't have good choice to transfer it as it is. So you have to convert it into non-reducing sugar, that is sucrose, so that it will not take part in the metabolism during transportation. Now you have to store it in the form of starch molecule. Now here you can see that in the C4 plant, phosphoenol pyruvate that is being captured as the C4 compound, and then it is moving to the bundle of sheath cell. So you have 2 different types of cell taking part in C4. Then you have a Calvin cycle, and as it is, things will go on. But you have to remember, you have to regenerate all the things and get the things back to the previous state. But in case of a CAM plant, you have different 2 conditions. Here C4 is oxaloacetic acid that will be converted to malic acid, and this malic acid will form the malate. So at the night time, what they have done, they have converted the CO2, because the stomata is open at the night time, so it will be captured as the C4, and it will be stored as a acid, malic acid and oxaloacetic acid, and the daytime this malate will easily release the CO2. So there is no requirement to open the stomata at the daytime. This is the simple way to understand. You can see the picture over here, maize and pineapple as an example. So most plant, tropical grasses like corn, sugarcane are C4, most plants are C3, and succulent plant like the pineapple and agave is a CAM plant. So now you can see here enzyme is a PEP case, PEPase is a PEP carboxylase, phosphoenol pyruvate carboxylase, and carboxylase means it will fix the CO2. Enzyme is of same, but here you have a Rubisco. You should give a slash, so it could be carboxylase and oxygenase, because it is bifunctional enzyme, and most energy efficient method is a C3, but it consume huge amount of energy, and normal case CO2 is the limiting factor, because it is not that much in atmosphere and loses water through photorespiration. That is the very risky thing, and lose water the least amount, that is in the CAM plant, and it is in between average of it. So now if you study the temperature versus photosynthetic rate curve, you can see the potential range for CAM plant. This is for the CAM plants. They normally stay in the dry place. Not only that, they also prefer to stay in a low temperature range, means normally like a 5 degree to 25 degree. And but in that case also their rate of photosynthesis is not that much high. So it is not that much abundant or economically, or in terms of a quantum yielding, it is not that much high. But if you study this part, the range of C3 plant is very good. Like if you take it from here, like around 5 degree to goes up to 37 degrees Celsius, as much the enzyme functions. So this is the potential range where temperature is somehow here, that is the top. So it is little bit less than 30 degrees Celsius, where you can see the highest photosynthetic rate in terms of micromole per meter square per second. But if you see, if it is high temperature range, like more than 30, close to 40 degrees Celsius, then the potential range for C4, that is very good and well enough and crossing the all bars. So that is represented in the blue color. That is how the leaf temperature and photosynthetic rate can be correlated in graph. This is another University of Zurich research that photosynthesis can be performed in an artificial way. So I think this is the end of the lecture. I think you have now clear idea regarding the C3, C4 and CAM plants and why they are existing, because they have a different geographically we are distributed, the atmosphere, the climate and the temperature, the dryness, aridity, all are different. So for that they have to adapt accordingly. So due to that, these are nothing but a adaptive behavior. And thank you so much. Please share and subscribe with your friend to get more like this video, and comment what more do you want.